Welcome to this explainer. Today, we're going on a seriously fascinating journey deep inside your own cells. We're going to explore the molecular foundation of human disease and, honestly, unravel one of the absolute greatest mysteries of our biology. You know, for a long time, we thought we pretty much understood how our DNA works. But modern pathology, well, it's revealed a breathtaking level of complexity that goes way, way beyond just a simple linear sequence of genes. So let's map out exactly how this hidden cellular world inside you actually operates.
Let's dive right in with a statistic that completely shifted the paradigm of modern biology. For decades, scientists focused almost exclusively on protein-encoding genes. But here's the wild part. That is a tiny slice, making up just 1.5% of our entire genome. That means a staggering 98.5% of your DNA does absolutely nothing to code for proteins. For years, researchers literally called this vast expanse junk DNA. But today, we know it as genomic dark matter, and trust me, it is anything but junk.
And that brings us to a rather humbling biological riddle. Take a microscopic worm. It's made up of fewer than 1,000 cells, right? But it has roughly 20,000 protein-encoding genes. Now, humans also have about 20,000 protein-encoding genes, and a lot of these proteins are actually recognizable homologs, meaning they're incredibly similar across both of our species. So, if we have essentially the exact same number of genes as a tiny 1,000-cell worm, and hey, no offense to the worm here, what makes us so vastly different? How on earth do we get human complexity from the exact same number of parts?
Well, the answer is brilliantly simple once you just shift your perspective a bit. The difference between you and a worm isn't really in the building blocks themselves. Proteins and coding genes are just the construction materials. They're the bricks and mortar. The real magic, the actual complexity that makes you distinctly human, lies in the architectural blueprints. That 98.5% of non-coding DNA, that provides the critical architectural planning, directing exactly when, where, and how those building blocks are put together.
So let's break down what this architectural blueprint actually looks like. There are 5 major classes of functional, non-coding DNA that regulate our gene expression. First off, we have promoters and enhancers. These act like landing pads for transcription factors. Second, chromatin organizing factors that physically manage the structure of your DNA. Third, non-coding regulatory RNAs, which we'll explore deeply in just a second. Fourth, structural regions, like the telomeres protecting the ends of our chromosomes. And finally, mobile genetic elements. These are literally jumping genes that hop around our genome. The whole shebang works together in this incredibly intricate dance to tightly regulate how your cells function.
Section 1. What makes us unique? DNA variation.
When we talk about what makes us unique, and also what makes us susceptible to certain illnesses, we really have to look at the sheer scale of our shared humanity first. Any two humans on Earth are typically more than 99.5% DNA identical. Actually, we're even 99% identical to chimpanzees. That means every single difference between you and the person sitting next to you, your eye color, your height, your unique disease susceptibilities, all of that individual variation stems from less than 0.5% of your DNA. It is a tiny, tiny margin creating literally boundless diversity.
Now, there are 2 really common ways that tiny 0.5% varies. On one hand, we have SNPs, or single nucleotide polymorphisms. Think of these as a single typo in a massive instruction manual. They're single-site variants, and we have over 6 million of them identified. On the other hand, we have CNVs, or copy number variations. If SNPs are single typos, CNVs are entire paragraphs or even whole pages that are either duplicated or totally deleted. They cover large, contiguous stretches of DNA, anywhere from thousands to millions of base pairs. Both of these variations, especially when they land in that non-coding regulatory dark matter, can directly alter your gene expression and heavily influence whether or not we develop a specific disease.
Section 2. Epigenetics. Above the genes.
So DNA sequence variation doesn't exactly tell the whole story. For instance, classic genetics cannot explain why identical twins who share the exact same DNA sequence can develop entirely different diseases over their lifetimes. To solve that conundrum, we look to epigenetics. The prefix epi literally translates to above. So epigenetics refers to heritable changes in gene expression that are not caused by variations in the underlying DNA sequence. It's this dynamic, highly responsive layer of molecular control that acts like a series of switches. It turns genes on and off without ever changing the underlying genetic code itself. To truly understand how these epigenetic switches work, you have to appreciate an absolutely incredible feat of biological packaging. If you took the naked DNA out of just one single human cell and stretched it end to end, it would be about 1.8 meters long. That's almost 6 feet. Yet, the cell has to cram all of that material into a nucleus that is only 7 to 8 micrometers in diameter. That is a microscopic space. It requires an insanely complex structural organization just to make it fit without tangling.
So how does the cell pull off this packaging, which we call chromatin? Well, the DNA wraps around central core proteins called histones. It's very much like wrapping a long wire around a spool of thread. But these spools aren't just for passive storage. They are highly dynamic. We have chromatin remodeling complexes that can physically slide these spools around to expose or hide certain genes. We have chromatin writers that add chemical marks to the spools and chromatin erasers that strip them away. If a gene is tightly spooled up, it's hidden, it's turned off. If it's unwound, it's exposed and turned on. This chemical marking system is incredibly specific. Think of these epigenetic marks as a cellular traffic light system. Histone acetylation, for instance, tends to act like a green light. It opens the chromatin up and increases transcription. DNA methylation, on the flip side, acts like a strict red light. High levels of it will completely silence a gene. There's also histone methylation and phosphorylation, which can either open or condense the DNA depending on exactly where the mark is placed. This intricate chemical tagging system dictates exactly what kind of cell a cell becomes. It's what decides whether a cell is a neuron, a muscle cell, or, unfortunately, a cancer cell.
And this leads us to one of the absolute most exciting aspects of modern medicine. When your actual DNA sequence mutates, it is incredibly difficult to fix. But, as our source material highlights, unlike genetic changes, many epigenetic alterations are reversible and amenable to therapeutic intervention. Because these are just chemical tags added by writers and removed by erasers, we can actually develop drugs to target that specific machinery. In fact, several drugs that inhibit DNA methylation are already being used in the clinic right now to treat certain types of cancer. It's a real game changer.
Section 3. The power of non-coding RNAs. Okay, let's look at another massive component of our genomic dark matter, non-coding RNAs. These are sequences transcribed from our DNA that never ever translate into proteins. Instead, they act as master regulators. First up are microRNAs, or miRNAs. These tiny molecules pack a massive punch through post-transcriptional silencing. Here's how it works. The cell produces a primary transcript, and then an aptly named, highly aggressive enzyme called dicer trims it down. This mature microRNA then associates with a multi-protein aggregate known as RISC. The microRNA acts like a GPS guide, leading the RISC complex directly to a target messenger RNA. Once there, it either chops the target up or completely blocks it from being translated. It's a highly targeted search and destroy mission. Interestingly, this is a very ancient system. Even bacteria use a primitive version of this exact machinery to chop up invading viral DNA. Now, beyond the micro, we also have the macro, long non-coding RNAs, or LNCRNAs. This is a huge, largely untapped universe of regulatory transcripts that are over 200 nucleotides long. By some calculations, there might be 10 to 20 times more of these than there are standard protein-coding mRNAs. One of the most famous examples of this is the XIS gene. In females who have 2 X chromosomes, one of them needs to be turned off to maintain biological balance. The XIS gene produces a long non-coding RNA that actually forms a physical repressive cloak over the entire X chromosome it was transcribed from, effectively silencing the whole thing. It is a stunning example of how RNA doesn't just carry messages, it performs some serious structural heavy lifting. So as we pull all of these different threads together, the core takeaway becomes incredibly clear. Modern pathology, the entire study of disease, is no longer just looking at a linear code of A's, C's, T's, and G's. That's old news. It is built entirely on understanding these complex, multidimensional molecular perturbations. To understand disease today, we literally must look at the dynamic epigenetic packaging, the chromatin remodeling, and the vast networks of non-coding RNAs that make up our genetic architecture. Which leaves us with this final, profound question to consider. We know for sure that much of human disease stems from the 98.5% of our genome that is regulatory. We also know that, unlike a hard-coded genetic mutation, epigenetic alterations in RNA regulations are dynamic and, most importantly, they are reversible. So if we can fully decipher this genomic dark matter and learn how to safely flip these biological switches, could we ultimately unlock the cures to our most complex, devastating diseases? It is a thrilling frontier in medicine, and one that scientists are actively exploring right now. Thank you so much for joining me for this explainer, and keep questioning the blueprints of your biology.